Good evening, and I welcome you back to our channel this evening. Today we're going to study an interesting topic for microbiology, and wetin we wan discuss the characteristics of prokaryotes, archaea and bacteria. We know say as we discuss, for some of our videos we don't discuss am say e get a method wey we dey take classify all the organisms wey dey on the earth, and one of those methods na the 5 kingdom method of classification. Now, for this 5 kingdom method of classification, one of the kingdoms wey dey very important to microbiologists na the kingdom Monera. Now, for inside that kingdom Monera, e get two major organisms or two major categories of organisms wey dey important, wey dey share this kingdom Monera. And those organisms, the first one na archaea, and the second one na bacteria, eubacteria or true bacteria. Now, these two organisms, these two categories of organisms, they been share many characteristics wey dey quite similar one to another. They been look alike. You get wetin we dey call RNA sequences. I'm going to discuss these ones for further videos. The RNA sequences wey dey for both archaea and bacteria dey very similar. They dey very alike in the sequences. So it will make those scientists to group these two organisms together for the same place. Now, e dey important to know say archaea, they no dey get cell wall, and cell wall na one very important characteristic wey bacteria get, but archaea dey no get cell wall. Wetin dey get na wetin dem dey call pseudopeptidoglycan. Na structure wey dey get, wey dey use help control themselves, maintain themselves, protect themselves for their environment. Na the structure wey dey get. Meanwhile, bacterial cell get wetin we dey call peptidoglycan cell wall, wey dey very important. Now, another thing to know na say a lot of archaea dey live for inside conditions wey dey very extreme. And wetin be extreme conditions? Many times, many organisms no dey like if salt too much for an environment, or if heat too much, high temperature dey, or if salt too much, or if high temperature dey, or if pH dey. Now, we go discuss all these things. These things na extreme conditions. You go see say many times, if our mama dem wan preserve many things, maybe meat for an example, if they wan preserve am, sometimes they dey use salt, rub salt for the body of the meat. Now, we never ask ourselves say why this thing dey fit preserve meat. Why be say if you rub salt for the body of meat, why e dey fit preserve that meat and that meat no go spoil? Now, we know say for anything like that to spoil, na say microorganisms dey act on that thing. They dey chop the nutrients wey dey inside that thing. Na wetin dey spoil am. Na wetin dey make food dey spoil. Na wetin dey make bread dey spoil. Na microorganisms dey do all those work. So anywhere wey salt dey, now one thing wey salt dey do na say salt dey draw water comot from the body of microorganisms. And if you draw that water too much, the cell of that microorganism go collapse, getting wetin we dey call flaccidity. That cell don collapse, and if the cell collapse, the microorganism no go fit maintain life again. So this one dey important. Now, make we discuss archaea wey we dey talk. Archaea been say they fit live for many conditions wey dey extreme. They get the capacity, they get the capability to stay for any environment wey harsh conditions been dey. Now, one of such harsh conditions na make salt content plenty for inside an environment. Now, archaea wey dey fit live for that kind of environment, we dey call them halophiles. Halophiles mean say they be organisms, they be archaea wey fit live for condition where the salt content wey dey that place dey very high, yet they go live for those conditions. Now, another one na some organisms, some archaea, they dey fit stay for areas or environments wey get very high temperature, very high temperature. And when temperature dey very high, those kind of organisms, we dey call them thermophile. Thermophile, they fit dey for those places where temperature dey very high. Places like where volcano bin erupt, for inside furnace, all these very hot places where fire dey, some organisms fit dey there. They go dey live, they go dey thrive, they go dey do whatever they dey do, their activities, they go dey run am for that place, even though say the place dey very hot. E no go kill them, no be say e go kill any other organism normally. Now, another thing na pH. pH dey very important. And wetin be pH sef? pH na measure wey scientists dey use check how much acid dey an environment, or base. You know say acid dey, the opposite of acid na base, or we dey call am alkaline. So if environment dey now, if acid dey very much for that environment, that environment go get very low pH. Na say acid don plenty for that environment. And if the environment get many alkaline materials or base dey plenty for that environment, you go see say the pH of that environment go dey very high. That pH e get scale, e dey between 0 and 14. So normally organisms dey like neutral pH. Many organisms dey prefer to stay for neutral pH, place wey no dey too acidic and e no dey too basic. They go dey thrive well. But we realize say archaea dey no mind whether the place dey very highly acidic or e dey very highly alkaline in nature. They go dey live there, they go dey thrive there. So we get name for them. The ones wey dey enjoy very low acidic pH, we dey call them acidophiles. And the ones wey like very high pH, wey be say that place go dey very basic or very alkaline in nature, we dey call them basophiles or alkalophiles. Now e dey very important to know all these terms. Now for science too, you know say science dey borrow English, and if you understand some things for English, you understand them for science. If you see the word philic, philic, e mean say that thing love that area or love that thing. If you see phobia mean say e no like that thing at all. So if you see for science, if they say something dey hydrophilic, e mean say that thing bin love water. If they say something dey hydrophobic, we know say hydro na water, e mean say that thing hate water. So when you say acidophile, extremophile, thermophile, you know say e dey talk say e love all those different conditions, and that one dey very, very important. Now a very well known member of the archaea na methanogens. Now methanogens dey be special organisms wey be archaea, and they dey special because their name na from methane. We know say methane na gas like our natural gas. So methane. Now why do they call these organisms methanogens? Na because those organisms, as they dey run their normal metabolic processes, they dey give out, they dey pass out methane gas from their body even as they dey run their normal metabolic processes. So na why scientists begin dey call them methanogens, say they dey generate methane from their body, and that one dey important. Many methanogens, they dey find them for inside the body of ruminants. Now wetin ruminants be? Ruminants na those animals wey get 4 stomach, animals wey dey chew the cud, wey dey vomit wetin they don chop, chew am again, chop am again, come swallow am, like our goats, like our sheep, like our cattle. We dey refer to them as ruminants because they get stomach wey get 4 chambers, 4 different chambers, and they dey vomit wetin they don chop, re-chew am again, come swallow am again. We find out say many archaea dey live for inside the body of many of these ruminants. They run all their different life processes for inside the body of these ruminants. Now archaea dey one side, the other side na the eubacteria or the true bacteria. Remember say I tell you say the true bacteria and the archaea, both of them, they dey share place together for inside the kingdom Monera. Na the 2 organisms wey dey inside that kingdom Monera. So e dey important to know this. Now the true bacteria or the bacteria, wetin dem get, wetin make them dey special? First, they get wetin we dey call 16S rRNA sequences. I been tell you say the archaea too, they get these RNA sequences. We go still discuss all these things further in this channel. We go take our time, discuss DNA, discuss RNA, discuss why they call some 16S, why they call some 30S, 80S, wetin all these things mean. We will calm down, discuss these ones for future videos. But for now, e dey important to know wetin bacteria carry. Now bacteria, they bin get peptidoglycan cell wall. So they get cell wall. That cell wall dey help them protect bacteria from all the evils wey dey the environment. Remember say na survival of the fittest. As you wan survive, the bacteria too wan survive. So that peptidoglycan cell wall dey help the bacteria to survive for inside harsh conditions. Now bacteria too don try, in as say e don conquer a lot of environment, and e dey many diverse environments. For some of the videos wey we don do for past, we don describe bacteria say they dey ubiquitous, meaning say they dey for different environments, and this one dey very true, and e dey important to know. We need to also know say bacteria get different ways wey dey take run different activities in order to keep itself alive. Some bacteria dey photosynthetic, meaning say they get chloroplast and chlorophyll for their body. They dey fit trap energy from the sun, come use am run the process of photosynthesis use am generate material wey dey go feed am, wey dey go feed on. Example of such bacteria na our cyanobacteria, dey dey run such process and e dey important. Other bacteria dey like some dey dey call dem chemosynthetic bacteria. These ones dey go use different chemicals such as magnesium or sulfur. Dey go carry those materials use am run different metabolic activities or reactions on top those chemicals, come produce wetin dey wan chop even using those chemicals. These ones dey important and we dey call dem chemosynthetic bacteria. Now we get bacteria wey dey heterotrophic. Those ones dey no fit produce any food, dey just dey like animals, so dey go go dey feed on different materials. Dey go dey, so if you see your eba spoil, na microorganisms or bacteria dey feed, dey fit feed on your eba, dey fit feed on your bread, dey fit feed on leather. Different materials dey, once dey don see sugar or see protein for inside that material, dey go begin dey feed on am, and na wetin dey make material spoil, na wetin dey make food ferment, na wetin dey cause different things for our environment. Now another thing to note na say many bacteria dey pathogenic in nature, meaning say dey dey cause many disease for the body of organisms. This one dey important to know and to understand. Many bacteria dey cause many different diseases, and as we dey discuss dey go for this our channel, we go discuss many bacterial diseases and wetin dey dey cause and how to guard against such diseases. But no be only trouble bacteria dey find o. Bacteria dey also exist inside relationship wey we dey call mutualism, or we dey say that, we go talk say that relationship dey mutually beneficial. Now wetin this one mean? Wetin this one mean na say bacteria fit dey for inside relationship wey be say no be harm, no be disease e dey cause, e dey help the organism, dey dey help one another. You ask me say which kind example I get. Now a very simple example wey dey na one bacteria wey we dey call bifidobacterium. Bifidobacterium, we dey find am for inside the belle, the stomach of children and some adults. Now wetin the bacteria dey do for that stomach? Two things now. You know say I tell you say mutually beneficial relationship na say you dey gain and dey also gain. Now that bacteria, the benefit wey the bacteria dey take na say e get home, e get habitat, e dey stay for inside the stomach and small intestine of human being, especially children. E dey plenty for there, so that place don be im home. E dey there dey enjoy himself. Now wetin be the benefit for the human being wey the bacteria dey live inside im body? The bacteria dey help to digest milk. Some people dey say some people dey lactose intolerant, dey no dey fit digest milk. Some of the things wey dey lead to that na say that bacteria no dey inside the body of that person. So if bacteria dey inside, if this bifidobacterium dey inside your body, you go fit digest milk very easily. If you drink milk, problem no go dey. People wey no dey fit digest milk, if dey drink milk now, dey go begin dey shit water shit. Dey no fit sit down for one place because their body no fit digest that milk. That milk don become dangerous to their body. If you check am, some of them, no be all of them, dey no get enough of that bacterium, bifidobacterium. So that one na mutually beneficial relationship wey this bacteria get with the human host. E get many other examples too wey we go dey discuss as time dey go. Thank you for listening to this class, and may una have a very good evening. Good night everyone.